Okay, hello and welcome to Chapter 7 webinar. And now this is the part 4 and it should be the final part of this chapter. And so I'm adding in a couple of things that I think are going to be very important, and you'll see that towards the end of this lecture. But this will have to do with lipids in particular, and then we're going to actually segue just a little bit back to the importance of sugars. And but you'll see, I don't want to give away the big reveal right until the very end. But I think that you'll appreciate this, especially for those of you that are continuing to want to pursue a nursing career. Okay, so here you go. We had mentioned yesterday about the differences between polar and non-polar molecules in their solubility characteristics. We talked about micelles, we talked about emulsification, and with those molecules, obviously you had polar and non-polar groups. And so we're going to be talking a lot about phospholipids here in this part 4. And one of the examples that is pretty common in foods is the phospholipid called lecithin. And so you see that you have these long chains of nonpolar molecules. So you have one in green, you have one in blue, and then you have these polar head groups here, one with nitrogen and one with phosphorus, right? So it's very similar to the triglycerides, where the triglycerides had 3 long chain nonpolar environments, but they were headed up by a polar group. For instance, it was at the head of that ester group, really with the in the triglyceride molecule. It's a very similar form here, except for without one of the nonpolar groups. So now you have 2 long nonpolar chains, or modestly long. I mean, obviously there's longer structures here, but relative to the head group here, which is polar. And so what's interesting, about this is that it is a yellowish fatty substance. It's in plants and animals. It's especially present in egg yolk, milk, soy, cottonseed, etc. It's also called amphiphilic or amphipathic. Either term can be used. We had introduced that previously as well. And again, it has largely nonpolar character, but the head groups do in fact help make this a bit more soluble. So it is an excellent emulsifier, and it's used as a food additive. So you can take a look at some different foods, and if you see that it might be made with lecithin, usually it's soy lecithin, and of course egg yolk naturally has it, then you're going to be able to see that this will lead to some emulsification. So this is one of the reasons, for instance, why the egg yolk, which is the yellow part, and is generally regarded as the quote unquote, less healthy portion of the egg relative to the egg white, if you will, does have that kind of emulsification looking property, right? It's all balled up within that egg. So this is just one of the examples of that. But let's talk about phospholipids with a cell membrane structure, okay? So again, we're going to be talking about amphipathic or amphiphilic, whatever term you like to use, or whatever term you end up seeing in your homework or even on the exams and such, or you just come across in the course of your research or readings or practice. Again, you have a non-polar fatty acetal and you have polar phosphate head group. So again, just like the triglycerides where you had several of these long chains, instead of having 3, now you only have 2, and instead you put on this organic phosphate head. And that then is the phospholipid. So again, 2 fatty acid tails as opposed to 3 for trigs. And this is really the general structure and what you're looking like. It's not that much more, differentiated from the trigs, with the exception of that there's not another tail. So how important is this? Well, our whole cell membrane layers are made up. They are comprised of phospholipids. And it's this is a nice structure of it. You see that in the bottom right, we have a cartoon of a cell with the cell membrane, with the cytoplasm present, and with the nucleus. If you were to blow up what that cell membrane looks like with this orange arrow that's leading to the top of the slide, you'll see that there's going to be a menagerie of things, right? So it's very important to understand that the phospholipid is always existing as a bilayer. So it's not just a single phospholipid layer. Instead, you have a bilayer, so with the non-polar tails oriented to the inside. This means that the polar head groups, which are denoted by these spheres, are going to be making contact with the cytoplasm within the cell, and then of course with the extracellular fluid, that circulates all about it. Now, as a part of what is in that cell membrane, you could easily see that you're gonna have to allow for passage in order to admit certain nutrients, including ions and stuff, or the shuttling of different products from within the cell to the to the extracellular space, right? Especially if this is going to be flushed with the blood flow and stuff. So this stuff is very important. You have a multitude of different functional components here. So you have peripheral proteins, you have cholesterol molecules, you also have the integral membrane protein, and you also have some carbohydrate side chains, and we'll be talking about that in just a minute. But you're definitely going to have some hydrophilic and hydrophobic regions, and all of this is so important. So remember that phospholipid layers of the cell membrane exist as a bilayer, not as a monolayer. They do provide the cell structure, just like my cells work to form that spherical type shape. Again, here the phospholipid bilayer helps to provide some kind of structure, some kind of form to the actual cell. And not only that, but it provides boundaries, regulates ions, proteins, and other components. So let's take a look at the phospholipid bilayer from like a cross-sectional view, if you will, maybe a two-dimensional view. This is just a blown up then cartoon of what that cell membrane would look like without any of the other components. But again, this just helps you understand about the differences between the polar and the non-polar regions inside and outside the cell. Again, it provides functional protein attachment, and it can also provide carbohydrate attachment, okay? So if you're talking about blood cells, like, I don't know, red blood cells, and we know that there are different types of blood types, well, you have all the different blood types, and that is going to be dictated by whatever sugar molecules, right, are attached to the polar head groups. And so that's one way of differentiating, certain blood, depending on whatever types of sugar molecules that have been functionalized to that red blood cell. So let's take a look at some of these. So we already had discussed the micelle previously, and that was used, for instance, to get rid of the dirt, right? So detergents and soaps can form that. And here you have this liposome, and then of course you have the bilayer sheet. The bilayer sheet we already are familiar with because we just introduced that with respect to the cell membrane. But a liposome is also an interesting structure, and instead of having a non-polar inner core, it actually has a polar inner core. And so it's very similar to the bilayer sheet, but instead of having a sheet, here you have an actual spherical structure. So what does this, what is this thickness? Well, it ranges about on the order of about 40 angstroms in the bilayer sheet. And that will be comparable, could be smaller, could certainly be larger if you're talking about the liposome. So, oftentimes with this shape on the left here, this liposome type of shape, this like this moiety, if you will, sometimes it's considered a vesicle. And so what would be some of those differences? Well, a liposome would be considered to be either natural or synthetic in origin. So liposomes are often thought of as being a potential carrier for drugs in order to be able to get into the body and then deliver the payload at the target site. And so the polar head groups in the middle here, obviously that would help to hold something that would be polar in nature, or be able to be responding to those polar head groups. So whether that's some kind of salt kind of structure, like maybe the drug is given in salt form, for instance, or maybe there's some pretty strong dipoles there. Either way, or even hydrogen bonding, all of that stuff can lend itself to being incorporated into the middle of the liposome. Vesicles typically referred to as an exclusively origin, natural biological origin. And so within, I show you an SAM here image on the top right. That's just a two-dimensional image of a cell membrane. That doesn't necessarily mean that you know, we don't know as much about, you know, what would be on the surface of this particular structure. I'm just showing you that as an example, but that would be an example of the bilayer sheet, going back to the dual layer here, this bilayer, and you can see how that would give rise to that membrane. You'll notice that the edges within that cell are a bit darker within that bilayer, and that's owing to the pretty polar head groups that are going to be residing within that structure, within that cellular structure. But here would be an example of where you have some functionalized phospholipid bilayers. Remember whenever we said that you could have sugars attached to that cell membrane, right? You can also have proteins as well. In this particular case, these are going to be some saccharides, and all this stringy stuff that is attached on the outside of this membrane here. And this is an SEM or a TEM image actually of the Bacillus subtilis, also known as hay. This is a type of bacteria, so it could be referred to as a common hay bacteria. You would see that the sugar molecules, which are the stringy stuff, are attached to this phospholipid bilayer. So as an example with the red blood cells, and they have the different sugar, you know, groups, which they do, you can imagine a very similar type of looking structure. Within the bilayer, okay, let's go back and now let's talk a little bit more in detail about the peripheral membrane proteins, the integral membrane proteins, the cholesterol, the channel proteins, the glycolipids, and the glycoproteins. So, you know, you have a variety of different functional groups within that cell layer that are giving rise to different properties. And I think I mentioned previously, the whole study of sugars on cell membranes had been poorly understood many years ago, but there's been a lot more research dedicated to it, and it's quite important because obviously we know that the sugars provide a nice sticky substance, and if you know there's any kind of opportunities to incorporate some of these sugar molecules, whether they're with proteins like the glycoproteins or whether they're with the glycolipids, then that's an interesting avenue of pursuit. So we know that attachment of certain moieties can then be had through sugar groups, right? They can be connected with proteins, and they can be decorated on the cellular surface. What about some of these peripheral membrane proteins? What are they about? Well, we know that they don't penetrate all the way through the bilayer. So what is their role here? Well, they work with the integral membrane proteins, and they do provide reversible attachment. So that's pretty interesting for uptake or elimination of certain components from within the cell. Whereas the integral protein, they actually just resemble a channel, right? Some kind of flue, right? That goes from within to without. And these proteins, they do close, they open as needed. It all depends on what the requirements are. So you have channel proteins, which are you know, quite similar to these integral membrane proteins. You have cholesterol molecules that also decorate within that bilayer. So this is a very active layer within the cellular structure. So quite interesting. I'm sure you'll get more information about that in some of your other courses, and I don't want to diverge too much from that. So here I wanted to talk a little bit about dental plaque. And I had previously mentioned to you that this is largely sugary in nature, right? That the starches that are produced, or the saccharides that are produced, you know, have analogs to what the plaque is in the mouth. And I want to visit this now because I think it's going to be important, because at the very end of this lecture, and this is one of the shorter lectures that we have in the series with this chapter, this will become a little bit clearer in terms of why I would even be talking about this. So we know that we need to clear dental plaque because, well, it's just nasty as those layers sit upon your teeth. I mean, we all know that prior to brushing your teeth, that sticky layer is just unhealthy, and it's just nasty, right? So we want to have a clean mouth. But here, let's just talk about some general observations. So all that sticky, nasty stuff in the morning whenever you wake up, especially if you've had a pretty sugary diet the night before, you're going to find that that this is synthesized actually by the bacteria that already reside within your mouth. Okay, so they just metabolize the sugars, and they really like sucrose. And what happens is that these bacteria express polysaccharides, and these are in the form of dextrans and levans. And so the dextrans are primarily comprised of glucose moieties, and whereas the levans is particularly comprised of fructose. So you know that glucose plus fructose gives you sucrose. And so the digestion of sucrose by bacteria can lead to these polysaccharides. We call them extracellular polysaccharide, sugars, really. And so I just give you an example here of dextran. Again, dextran is just a series of glucose monomers all attached. And I also show that you still have the animal carbons, as you know, from the alpha-1, 6 and the alpha-1, 4. This is the formation of those. And the alpha, again, refers to the down connection instead of the up connection, which would be the beta. And what you're looking at is then the 1 to 6 coordinated position, or the 1 to 4. So, there's varieties here, and there's multiple branching. So I think that that's kind of an interesting way to look at this too. This isn't just like those polysaccharides that we had talked about before in terms of cellulose or chitin, or just things like that. We have actually multiple points of bonding. Okay, so these are dextrans. With respect to the levans, well, that's a different beast, right? And here you have some connections, but they only are going to involve themselves with fructose, and you're going to have a number of chains here going. It looks like you have, carbon 1, 2, 3, 4, and 5. So you're going to have some interesting polymerization there. That's going to give rise to the levan structure. These ones are, in fact, gelatinous. I mean, you guys know this if you floss, or if the dentist or hygienist actually cleans your teeth. I mean, they'll know it. These sticky surfaces, okay, serve to support and sustain bacterial population and growth. Right? So functional attachment is so key. You can't colonize without having planted some kind of foundation, some kind of roots, if you will, in a given area. And that's some of the purpose of these polysaccharides. You start laying the groundwork to be able to incorporate more bacterial species. It could be similar species, and it could be a bit different, but generally the pathogenic species are going to prefer a more acidic environment, where the commensal bacteria are going to perform an environment that typically has a little bit higher pH. Not that it has to be hugely basic, but certainly around neutral pH. These saccharides, in addition to providing the attachment and being able to support a host of various bacteria, they can also be catabolized, okay? So in the absence of free sugar, then the bacteria turn to these stores. As we know, polysaccharides are often used as storage of glucose, or, well, in this particular case, glucose or fructose. And here, what happens is, especially the dextrans, because that is the glucose, and the bacteria especially prefer glucose, simple monosaccharide feedstock there, then they can access it. So for instance, if you see the importance of glucose within any kind of bacterial colony, here I show you this image, right? So this image, this was just a microscopy image taken all the way back in 1941. So investigations have gone back pretty far. You see that the S. salivarius colonies are definitely more amenable to a 5% glucose environment, as noted by all these spheres that you see in this left petri dish, relative to the petri dish on the right, which doesn't have any. And so, what is more important, glucose or sucrose? Always glucose. And do bacteria still have the ability to break down sucrose? Of course they do. But in this simple experiment, it's able to be very clearly identified the importance of glucose over sucrose. So This is a very colorful slide of then all the different bacterial species. Well, this is just a sampling of it, but this gives you some idea of what kind of supragingival plaque are living on our teeth. And this is just natural. So these samples were taken from people. They had their tooth surface swabbed for bacterial analysis. And whenever we say supragingival, so this is growing above that gum line. Okay, so this is decorated onto the surface. And they're able to identify basal layer plaque constituents. For instance, you have actinomyces species, you have streptococcus species, but then you also have additional species that are in the next layer, if you will, of the plaque. So you can have multiple layers of plaque, and those multiple layers can house different bacterial species. So you even have lactobacillus species, for instance, as noted in this image C of the second layer. So there's a variety of bacteria that can live on the tooth surface. If we're talking about, and that was above the gum line, if you're talking about below the gum line, then you have an even different makeup of bacteria. And so here you end up having, if you look, in addition to the actinomyces species, you can also have in this yellow and green images, B, C, D, E, and F. Take a look at all the different species that are now present here. So you have Synergistes, you have Fusobacterium, Tannerella, you have Treponemes. You have many different types. And so this begs the question, what is the difference possibly then in people that are more susceptible to having cavities versus those that are not? And this study was actually published more recently. It's called the Dental Plaque Microbiome in Health and Disease, and you can access this, of course, online. This is published in 2013. And if you look on the left, you have multiple colored bars, right? You have yellow, you have green, you have some blue, and then you have like this navy colored bars. And then on the right, you don't have much expression of anything. You have a little bit of orange, you have some red, maybe you have a little bit more blue than natural. So what does this all mean? Well, I show you here that you have more Streptococcus and Actinomyces species in the left, right? So you have some different Streptococcus species here on in the upper part. That corresponds with the red bar to having over 50% of all the counts are attributed to that species. You have some Bionella here too in the orange, and that's also pretty high. So the orange represents 5 to 10%. The yellow is representing anywhere between 1 and 5% of all of the bacteria that is comprised there. And then you have even lower amounts. So the ones that we have to start looking at is, well, the Streptococcus species, the Bionella species, the Campylobacter, and the Neisseria. All of those seem to be highly expressed in those that have active caries. And so it's interesting to note those differences. That's something that from a genetic perspective, and it could probably be modified with diet and just continual proper hygiene use and just dedicated compliance with a routine, whether it's food and or oral hygiene practice. But you're going to have limitations, because if somebody is naturally disposed to having certain type of bacterial species, well, then that's going to render a more risky environment just naturally. And so I use this as an example. Me myself, I've always had a pretty significant history of caries, whereas my brother has not. So is his ecological makeup different than mine? Absolutely. And what this study shows is that here, again, published in 2013, and this is in the PLOS One journal. You're free to look at it. You just click on these links and such, and you will actually, this shows that the ecology is certainly different. And so it's not just what you eat, it's not just what your compliance is. Sometimes we're just disposed to having cavities. Well, or I should say the risk of it. So, for what I do, of course I still eat sugar, but I just make sure that I'm pretty dedicated in my oral hygiene regimen. So I think it's pretty interesting. So, and you can see here that you have an overexpression of actinomyces, Veillonella, and Streptococcus all the way on the right. Now, I do want to mention that there are Streptococcus species that are commensal that are not pathogenic. Why I say that is because Streptococcus mutans is typically the target enemy number one, if you will, of saying, hey, this is the microbe most responsible for dental decay. Well, you know, it is once your teeth have been diseased to that point, then definitely Streptococcus mutans certainly comes to the forefront, but that's not one of the leading bacterial pathogens that are responsible for initial decay. Usually you have other things, including the actinomyces, including lactobacillus. And, you know, there's some other bacterial species that first get the tooth to a point where it is weak enough that all of a sudden the Streptococcus mutant species comes in and really starts to dominate that disease progression. So let's just keep that in mind, okay? So you have in this image right here, and I think that this is kind of interesting, and this is going to be really important because I want you to know how important it is to floss and brush your teeth or visit the dentist or whatever. So if we just look at undisturbed dental plaque, right? What does that look like? So this is just a cross-sectional image here of undisturbed plaque that had rested on dentin, okay? And this was, these are like dentin specimens that have been placed in a volunteer's mouth, and then after a certain period of time, that dentin specimen is removed from the patient's mouth and then it's analyzed. And this is what this example refers to. So the dentin surfaces are on the right in each image. So you see where in A and B, in the bottom right of each of those images, you have a horizontal bar. And the horizontal bar typically indicates of a size, and that size is noted as 10 microns. And near to that surface, you have then the dentin. So just regard the black regions here. I mean, if you want to, that interface there between this, you know, modestly colorful layer that immediately borders that black region, again, on the right-hand side in each image, would be the dentin. And again, dentin is the underlying material that supports enamel. So as your enamel continues to break down in a cavities related kind of scenario, the enamel dissolves away to the point where it leaves the dentin, and the dentin is softer, and that's really actually where Streptococcus mutans does its worst. It loves to feed on the organic matrix of the dentin layer. So this would be, for instance, a tooth. Whenever we say dentin sample, this would be a tooth that has been shaved clean of enamel. We're just looking at dentin, and we just want to see what is the effect on plaque on the dentin surface, especially if it's undisturbed. So after 24 hours, you can see in the figure A, so this is 24 hours, and B is 48 hours, you can see that there are some live clusters that are still interspersed within free spaces. Okay, so all of the greenish looking stuff, it may not appear green on your screen, but let's put it this way, the non-red material, that's kind of like intercalated throughout this image. You have a lot more dead material than you do live, it seems, okay? And you're looking at a plaque layer that is probably about 50 microns thick, right? So if we were to take those scale bars and just, you know, try to overlay that along that thickness, you can measure it. So it's about 50 microns thick of that plaque. It has been undisturbed. It's been in somebody's mouth for a certain amount of time. Wanted to see what happened. Well, all that red represents dead tissue. So that could be dead bacteria. That could be your own cellular tissue, the sloughing off that naturally happens on the inside of your cheeks, for instance, or your gums. Even some modest sloughing from the tongue, okay? But all that is dead material, dead organic material. If you were to let that go for another 24 hours though, look what happens. All the bacterial species, we're gonna look at figure B here now, you can see that there's a lot less red material. What this means is that the bacterial colonies that had already been colonized within that plaque layer, and again, it takes a little bit of time to recruit those bacteria species into a given plaque environment. Okay, again, cellular attachment is key. How it's doing this and how is all this plaque formed? These are all those polysaccharides, right? You're having the dextrans, you're having the levans, and all that is laying down the foundation, a blanket, if you will. And then there's invitation, but it'd be a very sticky blanket and it'd be obviously thick too, because that is what is going to be able to attract and retain those microbes that are going to be amenable to that environment. You know, what we have here then after 48 hours is a ton of live bacteria and not so much dead material. What happened to that dead material? Well, it probably got consumed. Okay. Now, what is the benefit then? So that was just an example of if you let plaque long enough in your mouth. So if I go back to that slide, this is an example of you can't reach certain areas of your mouth, whether that's because you're not flossing, whether it's because you have poor brushing, or whether you didn't brush at all, or whether you have existing maybe dental appliances in your mouth. So say, for instance, you have braces, you have retainers, you have any kind of structure within your mouth that prevents you from getting that area completely clean. Well, we call that plaque traps. And this plaque can actually set up in that trap then and cause problems because, again, the bacteria aren't just dying, they're consuming whatever is dying. And even after 48 hours, that plaque thickness is still pretty robust. And the colony, well, that's alive and healthy because they're feeding off of the dead tissue. So you may ask yourself, well, what happens if I can just use a mouth rinse, or what if I can't actually, physically remove that? Maybe it's too difficult to physically remove that from all of these traps. What happens if we just apply some kind of chemical treatment? Well, this is what happens. So figure A, then you have 24-old plaque. Again, we have the dentin on the right side in each of those images. The scale bar is still 10 microns. This was a control sample, right? So you have a 24-hour plaque sample. In this particular instance, you didn't have much dead material. And then what you do is you topically treat it with, in this particular case, chlorhexidine, which is known to be especially effective at killing bacteria. But what's really interesting is that after 24 hours, after an initial treatment here, this is for 1 minute, which would be typical, like some mouth rinses have this chlorhexidine. Usually this is a specialized mouth rinse. This isn't like the typical mouth rinses that you would have that might have fluoride in them or might not have fluoride in them that are for fresh breath. Usually these are suggested by your dental professional because you might need an additional layer of protection in your oral hygiene regimen. But anyway, chlorhexidine is one of the more prominent actives that we have that we can use to try to control microbial populations. But here's the limitation of it. You see that the chlorhexidine, although it had been applied for a minute and then the plaque had been monitored for 24 hours, look at how the dead tissue still resides on that surface. So within a size of probably, well, definitely no greater than 10 microns at that surface. I'm talking about the left surface in that figure B image. You're seeing that the penetration of chlorhexidine is not strong enough. It's not able to diffuse all the way through that 50 micron thick plaque layer, okay? And so with the dead tissue there, we already know from the previous slide that the live microorganisms, well, they're just gonna use whatever they got in terms of feedstock, okay? Including even their own kind that has subsequently died. So how important is it to get rid of plaque, especially if you have areas in your mouth that maybe harbor such traps that can lead to this plaque formation? And then these are the conditions which then ultimately provide an environment that can lead to cavities, even small cavities formed, like we call them white spot lesions, non-cavitated lesions, if you will, in the enamel. Well, all this plaque right here, this is so important to help get cleaned up. So again, what are the environments in your mouth that give rise to this? You have tight spaces between teeth. So if you have overcrowding in your teeth or a lot of overlap, and there's a lot of tight spaces, if you have orthodontia, if you have retainers, if you have anything that's going to be even crowns and stuff, especially if they're poorly fitting, but even if they are well fitted, it's really hard to get that surface really clean and all those surfaces inside really clean. But this is in essence, really, the underlying etiology of how bacteria can even start to colonize that area and create a problem in your mouth. So it's really important to clean that out. So how important? Even in 1977, and researchers published on this, and they said, look, all you have to do, as long as you can keep the teeth clean, as long as you can get the plaque out, you don't necessarily need to have another agent that can help strengthen the teeth, like fluoride, or even you don't even have to have something like a chlorhexidine to be put onto the tooth surface in order to help remove that plaque structure. All you really need to do is physically remove the plaque. But obviously, that cannot happen in all cases. And look at everybody's teeth, right? You may even have this in your own instance where you have orthodontia, where you know that you have tight crowding in your mouth. You know, the things you can do: floss, 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 brush, try to brush as well as you can. You might even try to get another type of irrigation instrument. You know, there's ones that can irrigate water throughout the mouth. You can even get like the vibratory toothbrushes that try to do a further, improved job of trying to loosen up all that plaque material. But that plaque material is sticky, so got to make sure that you have good bristles. You don't have to necessarily use hard bristles, though. Everybody, thinks, well, if you have harder bristles, is that going to do it? No, it's probably just going to beat the crap out of your gum tissue, and at worst, especially if you're a hard brusher, that might start to abrade a little bit of some of the weaker areas that you have in your enamel. So use a soft tissue brush, and floss. If you want to use some mouth rinse, then that's fine too, but I would use the mouth rinse first before I would brush, because we obviously know that mouth rinse is only going to get that surface layer, right? Just showing you those images there. So that would be one of the reasons why you want to, for instance, rinse before you brush. And also with the brushing, you need to allow at least 30 minutes for the actives, whether that's the calcium phosphate that might be present in the toothpaste, or whether it's just fluoride. Either way, you got to allow those agents to do their job. So now, this is the final reveal. Why am I even talking a lot about these plaques? And you might say, well, this is very interesting, and it is, but how does this apply? Well, this has been and could be, in the future, implicated in pneumonia, hospital-related cases. So with respect to the pneumonia, here you have normal air sacs, right, that are within the lungs. If you have them filled with fluid, then that gives rise to the condition which we call pneumonia. So how does this happen, and why does this happen? Well, this is really topical, because at the time that we're doing this webinar, obviously there's concern about a significant amount of people being admitted to a hospital with some kind of health-related disease, especially those that may be susceptible to the coronavirus. Well, as you are in the hospital, the #1 hospital-acquired infection is pneumonia. So that's a pretty interesting statement. This is from the CDC. They have reported this. So anybody who might be in there, even if you're not in there to counter the flu or the coronavirus or something else, if you're going to have an extended stay in a hospital environment, pneumonia is going to be the #1 risk. And obviously, if you have underlying medical conditions, if you're in a certain demographic, that's not going to be favorable for trying to fight this off, with your body, on your own without support from the hospital staff, if you will, then, or additional medications that you might take in a take-home environment, then this is going to apply. Okay, there's an 8.4 times increase in death risk whenever the hospital-acquired, #1 hospital-acquired infection, i.e. pneumonia, happens. 1/3 of patients, okay, who have the pneumonia will die, and anybody over 40 years of age are at risk. Is this an expensive, problem whenever somebody has pneumonia? Yes, it costs about $40,000. Okay, so even if the person doesn't, pay that directly, they may say, hey, well, I have good insurance. Well, somebody's paying for it. So whether it's the insurance company or whether it's the government, ultimately it ultimately comes back to somebody has to pay for this. So will you have increased premiums? Is that an increased burden on taxes in some other way that affects us? But most importantly, man, you're talking about a human being. And so how can you stave off pneumonia? And what is a really cheap way of doing this? And we would call this perhaps a minimal intervention strategy, just toothbrushing. We're not even considering just using, any kind of aggressive prescription strength toothbrushing, although some people with underlying conditions should typically be on higher strength toothpaste formulations, in my opinion. But we're just talking about brushing the teeth to disrupt the plaque. So for instance, the NYU Langone Center, 20 minutes after surgery, patients were ordered to brush their teeth. And here's another example. In the Sutter Medical Center in Sacramento, from May 2012 to December 2014, pneumonia went down 70%, okay, just by multiple brushing events. All right, so that's pretty amazing. And they compared that to the 2010 to 2011 example. And here, example 3, the Department of Veterans Affairs, this is in Salem, Virginia, that the study was done. Between October 2016 and December 2017, pneumonia was down 92% relative to 2010, 2011 models by 2 times brushing events daily. And do you know what's really interesting about this? And oh, by the way, you can read about this in the Wall Street Journal. Here's the issue. And if you want that, you can always write to me about that. I can share that with you. What's really interesting about this is that it wasn't dentists and it wasn't doctors that said, hey, I know a way of reducing mortality here in the hospitals, or reducing the risk of pneumonia contraction, basically, or infection, if you will. It was nurses. And so for those of you that are studying to be nurses, and if you're watching this presentation, and if you're able to look at this, and I gave you kind of a whole breadth of just different chemical, natures of sugars, I mean, obviously we talked previously in the other lectures in chapter 6 about all the differences and all the characteristics of sugars, whether they're monosaccharides all the way up to polysaccharides. Here I'm showing you that even in the oral environment, these polysaccharides can be produced. We see that the sugar molecules are incredibly important in functional attachment, right? Even to cellular layers. So we see that bacteria can be coated with these things. I showed you images of that. We talked about the phospholipid bilayer. And at the end of the day, this all comes down to how can we best care for the patients. And in these instances, you had nurses that were able to identify this problem, and obviously they probably they consulted with different hygienists and maybe dentists as well that are active in this stuff. But do they know about the importance of plaque? Not really, not to the degree that a hygienist or dental professional will, right? The dental professional already knows how important it is to remove the plaque that forms on the teeth. Nurses aren't necessarily acute to that because, well, that's just not part of the training. But here you get information that now can help you in case you find yourself one day working in this kind of environment. And one of the best ways to help try to keep a clean environment, a safer environment for your patient, well, that would be to brush the teeth. All right, that concludes the part 4 of chapter 7. And we will move on to chapter 8 in a future seminar. Okay, bye-bye.